hindi pa pala. Yan. So, we had your non-spore-forming gram-bacilli, right? So, 4 yung mga kailangan nating i-memorize class. You have your erysipelotic sociopathy, then you have your listeria monocytogenes, Corybacterium diphtheria, at saka Gardnerella vaginalis. What we need to do, class, in order for them to be differentiated is using your biochemical tools, of course. Diba? So, isa-isahin natin sila because there are some biochemical tools that will differentiate them, and we'll start with your erysipelotic sociopathy. With your erysipelotic sociopathy class, in a general characteristic in your morphology and staining, they are also known as your Chinese character bacillus. Why? Because in arrangement, they either produce a singly, short chains in or V-shapes. V-shapes, kaya nagkakaroon tayo ng Chinese character formation. Okay? In culture characteristic naman, class, take note with this one, they are alpha hemolysis. And we can only identify and observe the alpha hemolysis if kung may prolong tayo na incubation. So if you can observe on the picture, class, on your PowerPoint slide, you can observe here na meron tayong parang Chinese character letters. Yan. Okay? Yung parang madumi tingnan. And then, let's have your motility test for the identification of erysipelotic sociopathy. So with your erysipelotrics naman, class, okay, for motility test, madali lang yung principle natin. We allow the organism to move. Kaya diba your SIM is a semi-solid agar. Okay, so ibig sabihin, class, we allow the organism to move. So the positive reaction should be turbidity. Take note also with the stabbing. Okay, sa pag-stab natin, dapat yung depth natin, class, is 1/2 inch. If you can observe the picture, your erysipelotic sociopathy will produce a test tube brush appearance. Motility naman, class. Okay, so ito yung typical na ano natin. Appearance ng erysipelotic sociopathy natin. Aside from the motility test, class, using your SIM, we can also use your TSI via the use of your H2S production. Kasi your erysipelotic sociopathy, class, is an H2S producer. So let's try to decode the principle of your H2S production. So madali lang, class, kasi pagdating sa H2S production, what we need to do is to identify kung sino yung H2S source. So yung H2S source natin, class, is sodium thiosulfate. Tapos, your sodium disulfate, kakainin ni erysipelotic sociopathy, hindi kakainin ni listeria and other organisms. So, ang mangyayari, class, si erysipelotrix lang ang may kakayanan na kakainin si H2S source na sodium disulfate. Pag kinain na si H2S source, class, ano yung product? Diba? Every time na may kakainin ka, nandiyo kay ilibang, diba? And then, that is your sodium disulfate, class. In your sodium disulfate, is a colorless gas. So as a medical technologist, we cannot appreciate and observe a colorless. Tama? Kaya nag-a-add tayo ng H2S indicator. Okay. Sorry, class, hindi nag-a-add. Andun na yung H2S indicator sa TSI. So kakabit na si ferrous ammonium sulfate sa H2S gas monosodium disulfate. Kaya nagkakaroon tayo ng black precipitate, class. Kaya nagkakaroon tayo ng black precipitate. So for the characteristic of erysipelotic sociopathy, class, they are actually part of the normal flora of humans. They also carried by and causes diseases in animals. Okay, and that's the reason why, class, they are zoonotic. So with the animal exposure, class, usually with fish naman to, class. Kalang mga nasa palengke nga, nagkakaroon tayo ng erysipelotic sociopathy infection. And we call that one, class, erysipeloid. Sir, parang familiar, erysipeloid at saka erysipelas. Your erysipelas, ang causative agent natin, class, is correct. You have your Streptococcus pyogenes. Diba? So, ang ibig sabihin ng myeloid, class, makita gug kag mga word na myeloid. Okay? Ibig sabihin, anak class, kapariha, naay kapariha. Okay? So, erysipeloid is, nado siya kapariha, which is your erysipelas. So, they have the same clinical manifestation, but different causative agents. So, they are now called erysipeloid. Okay? So, erysipeloid is also known as your Butcher's disease. So take note with the two terms class, erysipeloid for erysipelosis. You have your erysipelas for your Streptococcus pyogenes. Then let's move to your Listeria monocytogenes. Di ba madali lang how to identify erysipelotoxicopathy? Pag nakita mo na non-spore forming siya na bacilli class, titignan mo kagad if kung may testubras appearance siya with your SIM. Second, if kung H2S producer siya. Okay? Then next, we will move to your Listeria monocytogenes. For your Listeria monocytogenes class, their appearance is almost the same with your with others except for erysipelotoxicopathy. Kasi si erysipelotoxicopathy class, mga V-shape sila. Pero kay Listeria, mga ano ni sila class, putot. Okay? So mga cocoidal in shape. Actually, with your Listeria monocytogenes class, they are always mistakenly identify as your S. agalactae. Because based on the other biochemical tests, and that includes first your hemolytic pattern. So yung cocoidal sila sa class in shape. Okay, tapos gram positive pa talaga. Okay, so ang hemolytic pattern pa talaga is beta hemolysis. So para talaga siyang S. agalactae. Okay, and then ang kagandahan ng ni Listeria monocytogenes class, we can use to cultivate that one at 4 degrees Celsius because they are cold-loving organism. Okay, kaya very common with food glass like milk, cheese, yung mga malalamig, yung ref nato because they can cultivate there. With the previous board exam, they usually ask class kung what culture media is appropriate for Listeria monocytogenes. Alam natin kaagad class na ang culture media is your McBride medium. But recently, class, tinatanong na if kung ano yung culture characteristic nila pagdating sa McBride medium. Kaya sinulat ko na lang din yung characteristic which is crushed glass appearance class. Ito lang naman yung importante class, crushed glass appearance naman siya. Okay, so take note with that one. Yan yung typical colony characteristic natin pagdating kay McBride medium class. Next, you have your SIM motility again. With your SIM naman class, the motility reaction is umbrella zone of growth naman. Kasi kita nyo to, okay? So, ang ganda ng picture, diba? So, this is your Listeria monocytogenes. Okay, so umbrella pattern naman class ang meron tayo. Okay, highlight natin umbrella pattern naman sa class. Okay, red alert tayo dito class that the motility pattern can only be observed at 35 degrees Celsius. Okay, so hindi pwedeng 37 kasi masyadong mataas class. So take note with this one, your erysipelotoxicopathy, although they have a test show brush appearance that looks like a positive result, pero negative yun sa class. Okay, your Corynebacterium diphtheria also are negative, and Gardnerella vaginalis are also negative. So your Listeria monocytogenes among all the 4 members of your spore-forming gram-positive bacteria, siya lang yung gumagalaw. The rest, hindi na sila gumagalaw, class. Okay? So, take note with this one kasi yung umbrella pattern can only be observed pagdating sa SIM. Tama? Okay, take note natin to class ha. Your umbrella pattern only with SIM. Tapos, your tumbling motility class can only be observed if kung meron tayong wet month. Okay, take note with that one. Okay, hindi tayo makaka-observe ng tumbling motility class pag gumamit tayo ng SIM. That's a joke. You can only observe the tumbling motility pag naka-wet month tayo. Next, you have your CAM factor. Ayun, lumabas na naman si CAM factor class. Christy Atkins and Match Peterson with your Listeria monocytogenes naman. Sir, anong kaibahan ni Listeria monocytogenes at saka ni S. agalactiae? Kasi yung kay S. agalactiae class, if you can observe here, the production is arrowhead. Kita niyo to? Arrowhead, di ba? Saglit. So, kita niyo to, this is arrowhead. Okay, this is a typical synergistic appearance of your S aureus, this one, to your S. agalactiae. Okay, kaya nagkakaroon tayo ng CAM factor. Okay, with your, ano naman class, tawag nito, Listeria monocytogenes, they use of your Rhodococcus equi. Actually, they can still use your S aureus. Okay? Pero they have this different pattern class. Kita nito ang pattern ng ano class, Rhodococcus equi to your Listeria monocytogenes, nagkakaroon tayo ng black. Yan. Okay, kita niyo yung black, mura siyang square, compare with the arrowhead appearance. Kaya nagkakaroon tayo ng black type, okay, hemolytic pattern of your organism class. Okay, take note with that one class ha, kasi halos magkahawig talaga si Listeria monocytogenes at saka si Streptococcus agalactiae. May CAM factor, beta hemolytic, cocoidal inform. Diba? So, ang kaibahan lang ni Listeria monocytogenes pag nag-reach siya ng CAM factor, ang appearance is black type hemolytic pattern. Kay Streptococcus agalactiae naman class is arrowhead appearance naman. So the rest of the organisms class, Clostridium, you have Corynebacterium dicteriae, you have fairlessopathy at saka Gardnerella vaginalis, they are CAM test negative naman. Kasi Listeria lang ang meron. Okay? Na di pa siya lang din gumagalaw, siya lang din ang may CAM factor. Okay? So as I mentioned earlier that your Listeria monocytogenes is found in dairy products sa class or contaminated food. They are colonizer of your animal, soil, at saka vegetative matter. So very prone for the environmental contaminant class. Endogenous strain naman class, colonized mother may pass organism to fetus. Ayan, same na naman, di ba? Familiar. You have the same characteristic of your correct class is agalactiae. Okay, so even like with the disease manifestation and saka mode of transmission, they share the same characteristic class. However, your agalactiae, very less for your food contamination compared with your Listeria monocytogenes. Your Listeria monocytogenes also class can survive at lower temperature, specifically 4 degrees Celsius. Kaya yung ginagawa natin class, pwede natin siyang incubate at to enhance the growth of your Listeria monocytogenes. Okay, for the virulence factor, they have your listeriolizing O class, okay, which is a hemolytic and cytotoxic toxin that allow the survival within phagocytosis. Yung ginagawa natin here, class, ganito. So this is your cell. Then this is your Listeria. Your Listeria is an intracellular organism. So yung ginagawa niya, class, nag-release siya ng listeriolizing O, LO, that will, tawag dito, inhibit the protein synthesis. So they will be able to survive inside the cell and replicate. So ganun din ka talino ni Listeria monocytogenes class. Okay? We also have your integrin. Your integrin will serve as a protein that will induce the phagocytosis. O, diba? Ang galing. Diba? Bakit class? Kasi si Listeria monocytogenes sa labas niya, nag-e-express siya ng internalin. Okay? Yung internalin kasi para siyang pampatawag sa mga immune cells. Kauna ko please. Yan, kainin mo ko. Yan, with the internalin. Kasi pag nasa labas kasi si Listeria monocytogenes, very prone with other factors that will try to destroy them. Kaya dapat nakapasok sila sa immune cells. Pag nakapasok na sila sa immune cells using internaline, ayun, hina-hijack ng Listeria monocytogenes, okay, using your listerolysine. And then also with the use of your ACT-A induces actin polymerization on the surface of the host cell, producing cellular extension and facilitating cell-to-cell spread. Hindi pa, grabe talaga si class, Listeria monocytogenes. Kasi for example, kung endothelial cell yung pinag-uusapan natin, class, with this one, so diba, si Listeria papasok kasi may internaline. Tapos pagpasok niya dito, release siya ng streptolysin O para ma-hijack niya yung protein synthesis. Tapos gusto niyang pumunta dito, class, with other cell, then gagamitan niya ng ACT-A naman, class. Yan, okay, have your ACT-A. Okay, facilitating cell-to-cell spread naman siya. Yan, so ang galing ni Listeria monocytogenes, class. Can you imagine? Diba, bacteria pa lang yun, class, pero ang dami nang alam. Diba? Pabida-bida talaga to si Listeria monocytogenes, class. May mga matatalino talaga mga bacteria, class. And that includes your Listeria monocytogenes. Your Staphylococcus aureus is one of the most brilliant bacteria also. Your Streptococcus pyogenes also. Pinakamagaling din in Listeria monocytogenes. We also have your siderophores, an organism capable of scavenging iron. Take note, class, that iron is essential for growth with the organism. So, nagkukumpit sila sa iron ng katawan. Okay? And then, from the human transferrin and of enhanced growth. For the disease manifestation naman, class, ayun, bacteremia naman yung common sa kanya, at saka meningitis din, class. Yan. And aside from meningitis, class, nagkakaroon din tayo ng encephalitis, brain abscess, at saka spinal cord infection. These are all included under your CNS infection. So since they are in normal flora, same, di ba, ng natin, Listeria monocytogenes. So yung early onset natin, nagkakaroon tayo ng granulomatosis infectisecticum. Ang naman ito, class, English version naman ito, class, is stillbirth or namamatay yung baby sa loob ng katawan ng mother. And of course, same ng Listeria monocytogenes natin at saka Streptococcus agalactiae is bacterial meningitis. So, sana ma-appreciate ninyo si class, Listeria monocytogenes, na almost same sila ni Streptococcus agalactiae, class. Even with the virulence factor and also the disease manifestation, the culture characteristic are almost the same. Kaya, we need to be careful in identifying your Listeria monocytogenes from your Streptococcus agalactiae. So we are done with your Erysipelotrix at saka Listeria. Then we will move to your Corynebacterium diphtheriae class. For your Corynebacterium diphtheriae class, the morphology is in same with your V-shape, kanina with the Erysipelotrix. They are also known as your Chinese character appearance pa rin. Okay? And then, kasi class, yung palisading na natin, appearance of this one, kasi nagkakaroon tayo ng pleomorphic, is during the multiplication of the bacteria class, nagiging clingy yung mother cells. Hindi niya totally nire-release yung daughter cell niya. Kaya nagkakaroon tayo ng V-shape. So, as I mentioned that one earlier also with your Corynebacterium diphtheriae, diba? That their granules is called your Baines-Ernst granules or volutin naman, guys. Okay? So, Klebs, Loeffler, Bacillus is the another name. For the culture characteristic naman, class, take note with the blood agar media, meron tayong tatlong characteristic. Pero yung lumabas sa board exam class, you have your metis. Sa metis, based on the culture characteristic of 5% blood agar, it will produce a fried egg appearance It will produce a fried egg appearance. A. Gravis, B. Metis, C. Intermedius, then D. None of the above. Yung sagot naman natin ng class is Metis na kaagad. Kaya, kinhighlight ko na lang din yung iba. You have your Gravis at saka Intermedius. Baka, yun naman yung itanong, diba? Hindi Metis. So, dapat ready na tayo, class. Next, you have your Loeffler's serum. Okay, diba? Nabanggit ko naman to sa inyo class, Loeffler's agar or Loeffler's serum. Or other name, you have your pie agar. Okay, the Loeffler's serum naman class, ang gamit naman natin dito, diba, is your horse. It enhances metachromatic granule formation of your plumorphism. We also have other culture media class to observe the colony of your Corynebacterium diphtheriae. Then you have your potassium tellurite medium. The colony characteristic is gray to black colonies because, take note class, meron siyang tellurite. Pag may tellurite yung mga agar class, always expect that the color colony is black. And then, good thing about your potassium tellurite medium, it will also produce a specific hallmark, other characteristic of bleach-like odor class. Okay, bleach-like odor. Okay, next you have your Tinsdale medium, which will also produce a black colonies with yellow halo. And also you have your cysteine tellurite blood agar or CTBA. Okay, black or brownish colonies with halo class. Take note that your CTBA is a modification of your Tinsdale medium. Okay, containing sheep red blood cells, bovine serum, cysteine at saka potassium tellurite. So yung kaibahan lang ni class ni CTBA sa Tinsdale medium class is with the inclusion of your cysteine. Next, you have your electes. I think it would be, I think you can easily understand the electes principle because tapos na kayo mag-immunology. So yung principle lang naman ni electes class, na-identify natin if the Corynebacterium diphtheriae is producing a toxin or not. Kasi meron tayong mga non-toxin producing Corynebacterium diphtheriae, meron din tayong mga toxin producing Corynebacterium diphtheriae. So to differentiate them class, we can use your electes. Your electes kasi is an in vitro. Ibig sabihin class, gumagamit tayo ng culture to identify that one. So yung ginagawa natin class, naglalagay tayo ng, sorry, where is my, wait lang class, di ko makita yung ano ko. So you have your strip here, this one, a test strip of filter paper containing your diphtheria antitoxin. So, ibig sabihin, class, eto, meron siyang antibodies. Okay? So, pag meron siyang antibodies, class, okay, place in the center of the agar. Okay? Then, you have your strip, mag-strip ka here. Then, 1, for example, this is your negative control. Then, 2 is your positive control. 3 is your unknown, non-toxigenic. And then, 4 is your unknown toxigenic. Pag nagkakaroon tayo ng line of precipitin, okay, it means that the organism is identical and or positive. So one best example to understand that one class is this one. So if you can observe here, this is your negative control. Diba? If you have your negative control here, sorry class, positive to, diba? Positive. Then may positive tayo, tapos may test control, may test strain, tapos may negative control. So, you will have a pattern of identity kasi yung positive control mo at saka test strain, nagkaroon siya ng curve. Okay? So, ibig sabihin, class identical. Kung ang positive control natin is toxigenic Corynebacterium diphtheriae, then the testing is a possible toxigenic Corynebacterium diphtheriae. Bakit? Nagkakaroon sila ng pattern of identity. If kung ano naman class, of course, di ba, ang test strain naman natin, meron siyang antigen, toxin here. Pero yung negative control, wala kasi siyang toxin, di ba? Kaya wala siyang pattern of identity. Okay? So, with the test, nagkakaroon tayo ng positive line of identity and negative no line of identity. Okay? So, the principle of your elect test always falls under your precipitation test. And you know that one, I believe, in your immunology. Kaya mas madali na lang nyo siyang naiintindihan. Tanong dito, okay lang tayo dito, class, with the elect test? Can I have a raise of hand if kung okay lang tayo? Okay, thank you. And then, for your Corynebacterium diphtheriae class, they are colonizer of human nasopharynx, but only in a carrier state. And then they can actually produce a bull's neck appearance class or swelling. This one. Sorry. Characteristic. Kasi ang ano natin is with the nasopharynx yung colonizer natin. Then for the direct contact since nasopharynx, pwedeng exposure to respiratory droplets. Pwede ding exudates from cutaneous lesion or exposure to inanimate object. So one of the virulence factor class of recurrent bacterium diphtheria is the diphtheria toxin. The diphtheria toxin will destroy the host cells, specifically on the neck part. So, dinidestroy niya yung nasa pharynx class kaya nagkakaroon tayo ng swelling. Okay, that's the function of your diphtheria toxin. Okay? So if kung nade-destroy na yung nasa pharynx natin class, nagkakaroon na tayo ng respiratory diphtheria. Okay? So which will produce an exudative membrane that will cover the tonsils. Okay? And we call this one uh, pseudomembranous pharyngitis naman, class. Okay, sulat ko muna here. Sulat ko na lang sa ano natin. You have your pseudomembranous pharyngitis naman, class. Okay, pseudomembranous pharyngitis naman siya. Okay, so yun yung condition associated with your Corynebacterium diphtheria class. So now we are on the last part of your non-spore forming organism, okay, which is your Gardnerella vaginalis. We started with your erysipetalixopathy, that your erysipetalixopathy is a TSI, H was producer, test tube brush appearance. Your listeria will produce a cam factor block using your Rhodococcus equi. And then there are motile compared with others. Then you have recurrent bacterium diphtheria, pwedeng electes, and other culture media that produce a jet-like colonies or jet black-like colonies. So we'll move to your Gardnerella vaginalis naman. Kasi your Gardnerella vaginalis, if you can still recall in your clinical microscopy, lumabas ito because your Gardnerella vaginalis is associated with your vaginosis. And using your histopathology class, we can observe clue cells. A clue cells class, as you can still recall in your clinical microscopy, it's a phenomenon where the organism will surround a certain epithelial cells. So if you can observe your class, meron kasi tayong maling connotation. Kasi pagdating sa clinical microscopy, always na dinidiscuss nila na ang ano daw, yung clue cells, we can observe that one using the normal na microscopy. Like for example, nagpa-urinalysis, nakakita ka og epithelial cells surrounding with the bacteria, then you call that one clue cells. No, because the clue cells can only be observed if you have a histopathologic examination. So if you can observe on the picture, dapat nakastain muna siya, class. Okay? So red alert tayo here, Gardnerella species has a gram-positive type of cell wall. However, the peptidoglycan layer is very thin. Kaya nagkakaroon tayo ng false negative result class. Kaya we always classify your Gardnerella vaginalis as a gram-neg and or gram-positive. Then these cells is a large squamous epithelial cells with your observation of your glue cells. Uh, gram positive or variable because of the peptidoglycan, and it is the causative agent of your bacterial vaginosis. And then lumabas din to sa board exam class with bacterial vaginosis. Okay? Yung bacterial vaginosis naman class is the interpretation daw at saka ano yung test name na ginagamit natin. So nilagay ko na lang here class, hindi pa pala, excited pa pala ako class. Pero ito na lang kasi i-explain ko na lang siya. Malayo ko pa pala class. Ito. You have your Nugent scoring system for your vaginosis class. Yung tinanong naman sa board exam is the interpretation. If your Nugent scoring system daw score is 4 to 6, what is the interpretation? So the interpretation class is intermediate for bacterial vaginosis. So nilagay ko na lang din yung complete na interpretation class 0 to 3, 4 to 6, at saka 7 to 9. Normal vaginal microbiota pag 0 to 3, 7 to 8 naman class is bacterial vaginosis. For the laboratory diagnosis naman class, let's start first with the culture characteristic. So with the culture characteristic naman class, although the 5% culture agar is not very reliable, pero majority of the culture characteristic of your Gardnerella vaginalis is pinpoint colonies. With the hemolytic pattern naman class, they are non-hemolytic. Okay, take note with that one class, they're non-hemolytic. Okay, and your Gardnerella is easily inhibited with your SPS class. So, ano yung ginagawa nga natin pag nai-inhibit siya with your SPS? Correct class. Pwede tayong mag-add ng 1% na gelatin. Okay, correct. Okay, tapos another specific culture media for your Gardnerella vaginalis naman class, you have your human blood bilayer twin 80 agar, which is I was able to discuss that one earlier with the culture media. Pero usual na tinatanong class is kung ano daw yung hemolytic pattern natin pag nilagay natin sa human blood bilayer twin 80 agar yung organism na Gardnerella vaginalis. The correct answer is beta hemolytic naman class. Okay, take note with this one, beta hemolytic sila, Gardnerella vaginalis. Next, you have your urease production naman class. Ito, madali lang yung urease production kasi tinitignan daw natin ang organism if the organism will release urease. Sino yung organism natin class? Correct, Gardnerella vaginalis. So if kung meron daw tayong Gardnerella vaginalis class, your Gardnerella vaginalis will release an enzyme urease that will hydrolyze urea to produce ammonia. Okay? So since the urease is this one, pink color. Sorry class. Ito yung original color niya, yellow. Okay, or orange. Tapos kung positive with ammonia, it will become alkaline. Kaya nagkakaroon tayo ng pink or intense pink color naman class. Kaya kagandahan ng urease production class is that your urease production, si Gardnerella lang yung positive class. Negative si fairlessopathy, listeria monocytogenes at sa Corynebacterium dictatoria. Paki-highlight na lang din class na pwede din nating i-consider si Rhodococcus species as urease positive class kasama ni Gardnerella vaginalis. So your Gardnerella vaginalis, as we all know that they're normal flora of your vagina. That's the main reason why class the women use your pH care, diba? Because your pH care will try to neutralize. Why? Because you have a normal flora that produces acidity. That is your Gardnerella vaginalis. Okay, in order for you to control and produce a disease called vaginosis or bacterial vaginosis. So, dapat makontrol ninyo yung pH environment. Okay? And then, mode of transmission, of course, endogenous lang sa class. Okay? So, sa inyo lang din. And then, you have your virulence factor, uncertain. Ayun, plus factor yan sa atin kasi wala tayong kailangang i-memorize. And then, the spectrum of disease and infection is bacterial vaginosis. And we were able to discuss that one earlier. Ito. Okay, you have your Nugent scoring system and interpretation of your Gardnerella vaginalis. So now let's move to the last part, okay, of your gram-positive, which is your uh, spore-forming gram-positive bacilli. Okay, so if you can observe on the picture, you can observe that the gram reaction is still gram-positive. The shape is bacilli. Then how will you observe? Of course, check for the spore. And then next, how will you check? How will you proceed again? You will check the oxygen requirement naman, class. Diba? Kasi kanina hanggang check the spore lang tayo. Pag may spore siya, anong next? Yun yung next na tanong, diba? So, oxygen requirement naman, guys. Yung oxygen requirement, meron tayong dalawa, aerobic at saka anaerobic. With your aerobic naman, class, dalawa lang yung kailangan natin i-discuss. You have your Bacillus anthracis at saka Bacillus cereus. Then for your anaerobic spore-forming gram-positive bacilli, madamay-damay konti, you have your Clostridium perfringens, Clostridium botulinum, Clostridium tetani, at saka Clostridium difficile. So let's start with the morphology and staining characteristic of your Bacillus anthracis and Bacillus cereus. Yung gusto ko lang i-highlight dito, class, is with your Bacillus anthracis, they form a square shape naman, class. Okay, square-ended naman siya. So kung i-drawing nato sa class, inana siya gnawong. Okay, square-ended man daw siya. Okay, and then gram-positive or gram-variable bacilli found singly in or chain. And then, meron daw siyang spore na nasa gitna. So, ganun yung itsura niya, class. Okay? So, dakun-dako na to kay putot man ni siya, class. So, square-ended. Sorry, class. Ayan. Huwag niyo akong i-judge. Square-ended. Ayan. And then, central siya. Okay? Then, the spore orientation gives the appearance of a bamboo-shaped rods. Ayan. Kasi, pwede silang magdikit-dikit, class. Para siyang bamboo-shaped appearance. With your Bacillus cereus naman, class, rounded end siya. Kung rounded end siya, class, ganito yung itsura niya. Sa end niya, pa-round. Yan. Tapos, yan. Round na naman siya. Sa end. Tapos, saan daw yung niya? Terminal or subterminal. So, either dito sa subterminal or sa terminal na end. Dito na po. Okay, so that's the difference between your Bacillus anthracis to your Bacillus cereus. With the naman, class, culture characteristic naman, class, you have here hallmark colony characteristic of your Bacillus anthracis is that aside from they are non-hemolytic organism, class, they will produce a medusa head or comet tail appearance. Okay? And also, although wala akong picture with the medusa head, class, ang meron ako is the beaten egg white consistency. Okay? This one is this one. Baka yung parang nagbatil ka ng class, egg, na white egg. Ito yung itsura niya. Okay, that is a hallmark characteristic of a hallmark colony consistency of your Bacillus anthracis. And next, you have your culture characteristic. So we use the culture media bicarbonate agar. Okay, take note with this one, class, kasi if you want to observe the spore-forming production of your Bacillus anthracis, we can use your bicarbonate agar. Your bicarbonate agar will induce the capsule formation. Okay? We also have your PLET, polymycin, lysozyme, ADTA, talus acetate. They will produce a creamy white doom circular. And then, yung pinaka-importante naman dito, class, is the growth. Pag may tumubo sa PLET, okay, either with any characteristic, class, it's indicative that the organism is Bacillus anthracis na. And Lastly, with the culture characteristic classes by the use of your egg yolk agar. Okay, your egg yolk contain your lecithin, sorry class, lecithin. Your egg contains your lecithin, and then the bacteria contains your lecithinase. And then, if the bacteria produces an enzyme called lecithinase, it will degrade your lecithin. Yan, forming a opaque zone around the colonies. Yan. Okay, so you have your egg yolk agar. Then next, you have your Bacillus cereus naman class. Okay, we're done with your Bacillus anthracis kanina, and then you have your Bacillus cereus. With your Bacillus cereus naman class, we can still use only with your 5% blood agar. And the culture characteristic class is frosted glass appearing colonies naman siya. Hemolytic pattern is beta hemolytic. You can also use your egg yolk agar class with your Bacillus cereus. Compare with yung Bacillus anthracis, ang daming kailangang culture media, mag-plet ka pa, diba? May egg yolk agar ka pa, may blood agar media ka pa. Pag culture characteristic na Bacillus cereus, pwede ng 5% agar media yung identification mo. So differentiate naman class your Bacillus anthracis to your cereus, you can use the following test. So for catalyst test naman class, all of the 2 is positive. With the motility naman class, si Bacillus cereus lang yung gumagalaw, non-motile si anthracis. Pag string of first reaction naman class, positive si Bacillus anthracis. Your string of first is also observed and used with your Vibrio class. Then you have your gelatin hydrolysis class na you have your positive for Bacillus cereus. So si Bacillus cereus class kumakain ng gelatin. Si anthracis hindi. And of course, you have your lecithinase production. Ayun, diba? Tinitingnan natin if the bacteria is able to produce an enzyme called lecithinase. And we can observe this one using your egg yolk agar. So, in your egg yolk agar, your egg yolk agar contains your lecithin, which is the substrate of your lecithinase. So, lecithinase is an enzyme that is produced your bacteria. So, kakainin si lecithin. Kaya nagpo-produce tayo ng halo or opalescent naman class. Halo. Opalescent halo surrounding the colonies. Ang kagandahan nito class that your Bacillus cereus is, anthracis is positive, your Bacillus cereus is negative. Then, let's move to your Bacillus anthracis class with the reserva. So, your Bacillus anthracis or BK2, so it can be found in environment and very common in your soil. Yan, contracted by various herbivores or mga metals. Yan, or mga, di ba yung mga nails, lansang, pag matunog ka, ayan. So, very high tendency of acquiring your Bacillus anthracis. Then, for the mode of transmission glass, direct contact or animal tissue or products such as your wool or hair, kasi nagkakaroon tayo ng wool Salter's disease. Wool Salter's disease naman kasi for inhalation of your spores. Nagkakaroon din tayo ng food poisoning glass with the ingestion of your contaminated meat and or with the multiple use of your injection using contaminated drugs. Kaya nagkakaroon tayo ng anthrax. Okay, di ba? Remember with the disease anthrax, the causative agent is Bacillus anthracis class. For the virulence factor of your Bacillus anthracis class, take note with this one, they are encapsulated and therefore the component of their capsule is polygamma diglutamic acid class. Take note with this one kasi lumalabas siya sa board exam at saka pre-board exam class, polygamma diglutamic acid class or PGA. Okay? And we also have 3 protein and exotoxins. So let's start with the basic one. Ito yung pinaka-requirement sa lahat. Dapat muna meron kang protective antigen or PA class. Your PA or protective antigen will serve as a necessary binding class. Okay? Kung hindi mag-bind si edematous factor at saka si lethal factor class kay protective antigen, parang wala lang din siya. Okay? So, anong ginagawa, class? Pag meron kang protective antigen plus your edematous factor class, it will cause edema. Diba? If alone, non-toxic daw siya. Pero pag si edematous factor plus your protective antigen, it will now cause your edema. Pag si lethal factor naman, class, dumikit kay protective antigen, it can cause death. Okay? So this is very important. The three virulence factor is very important in causing the disease, specifically with your anthrax naman, class. So as I mentioned, we have three different types of your anthrax. So you have your cutaneous anthrax, diba? So it will start with the here, a ulceration. This one is ulceration naman, class. Then after ulceration is with your black escar. Okay, this one is your black escar. Your Bacillus anthracis is under your bioterrorism organism. So dapat highly ano siya, class. Tawag ito, dapat ma-notify ka agad yung government if we were able to identify your Bacillus anthracis. And also, you have your pulmonary anthrax class. Okay, as I mentioned earlier, we have your Wilson's disease. You also have your gastrointestinal anthrax kasi, di ba, meron tayong ingestion of your meat. And then you have your injectional anthrax kasi by the use of your intravenous injection. Compare with your Bacillus cereus class, this is more on food-associated diseases. Your Bacillus cereus is ubiquitous in nature, may transiently colonize skin or respiratory tract. And nakukuha lang natin to, class, using tawag nito, poor preparation of food, specifically fried rice. And sino yung mahilig sa fried rice can actually cause toxins. And we have two toxins, class. You have your enterotoxins. Okay, ibig sabihin, intestine toxin. A toxin that will destroy your intestine. And then you also have your pyrogenic toxin. Pyro, ibig sabihin, fire. And then genic meaning producing fire. Okay, so the fire there, class, is not literally the fire but fever. So ito yung toxin that can cause fever, class. Yun yung tawag pyrogenic toxin. So to differentiate the two types of your Bacillus cereus toxins, either it is diarrheal or emetic. So we have the following parameters. So with the incubation period naman, class, of your diarrheal, it's more longer compared with your emetic. Pag sinabi nating emetic, class, is more on vomiting naman siya. Okay, very common. Emetic meaning vomiting. Diarrheal meaning diarrhea. Diba? So yung food implication naman, class, pag diarrheal, pag yung nakain mo meat products, vegetables, sauce, diarrheal type siya. Pero pag fried rice or boiled rice, then emetic siya. For stability to heat, of course, stable si emetic na type. Bakit? Kasi sa fried rice, 'di ba, kung nag-heat up ka, namamatay yung organism, but that's poor does not. So that's the reason why it can cause food poisoning. Compare with the diarrheal na part is tawag nito, madali lang din silang mamatay, class. Yung organism. Stimulation of your adenylate cyclase CCAM system, class. Yes, with your diarrhea. Bakit? Common with your diarrheal part. Okay, with your CCAM, class, ito ganito kasi siya. Like for example, ginasipon ka, you have your diarrhea, there is an increase of your CCAM. Okay, your CCAM, pag nag-increase kasi si CCAM, class, this is increase of water secretion and then electrolytes. So kung ginasipon ka, class, ibig sabihin you have an increased CCAM, and that is due to your organism. And can we push, class? Okay lang ba? I-push na lang natin to para one recording lang. And,
Yes,
Yes
sir.
po, sir.
okay,
Okay lang,
sige.
sir.
Sir.
Thank you. Tapos, konti lang na lang man tong mga anaerobic organism. Okay? So, you have your Clostridium perfringens, Clostridium botulinum, Clostridium tetani, at saka Clostridium difficile. Okay, in order for us to differentiate that one, either we can use your motility, lecithinase, lactose, lipase, at saka glucose. So these are basic biochemical tool to differentiate the four. And we will try to describe each naman class. So, good thing about the culture characteristic class is meron silang mga kukonting pwede nating magamit to differentiate. So, let's start with your Clostridium perfringens. With the staining characteristic naman class, they are boxcar shape naman. Di ba para siyang din, yung boxcar kasi at saka yung box kanina, whether Bacillus anthracis, almost the same. Pero the problem with your Bacillus anthracis class is that they are aerobic. Your Clostridium perfringens, they cannot survive in a presence of oxygen. So if you culture the media in an anaerobic condition and you observe a back shape characteristic, then that is most probably Clostridium perfringens. Okay, and then they also have the same characteristic of your Bacillus anthracis with the central to subterminal. Although your anthracis is only central, but with your Clostridium perfringens, pwede din siya mag-subterminal. In a real-world class, the Clostridium perfringens spore production is very difficult to observe. So, minsan nga, wala. Okay, so what you need to do is rely on the anaerobicness of the organism. Kasi pag in-incubate mo siya anaerobic, then therefore, wala ka na talagang ibang choice kundi yung apat na lang na mga Clostridium species. You can also use your blood agar plate, of course. With your blood agar plate, it should be incubated anaerobically. Kasi pag hindi mo yan siya in-incubate anaerobically, class, ibang organism yung tutubo. Possible your Bacillus group. Okay? Good thing about your Clostridium perfringens, pag siya, class? Pag na-incubate mo siya at anaerobic condition using your blood agar plate, they will produce a hallmark hemolytic pattern, and we call that one double zone of beta hemolysis naman, class. Okay, double zone of beta hemolysis naman yung meron tayo. Okay? Ayan, pangatlo na lang. Pangatlong recap na naman to, class. Okay, have your Clostridium perfringens. Okay, pero ang kaibahan ni Clostridium perfringens, class, is reverse camp siya. So, ang ibig sabihin, sulat ko dito, class, para mas maintindihan ninyo, you have this one. So, you have your camp factor. You have your camp factor, right? For your camp factor, for your S-aga, you have your camp factor for your listeria mono. You have your reverse camp for your Clostridium perfringens. For your S-aga class, ang gamit natin here na perpendicular is your S aureus. With your mono na camp, ang gamit naman natin, class, is Rhodococcus equi. With your reverse camp naman, class, ang gamit natin is your S agalacti. Okay? Tapos, yung organism naman natin here, class, is your S agalacti. Yung organism naman natin here is your listeria monocytogenes. Tapos, you have your Clostridium perfringens as this organism. Next is with the positive reaction, arrowhead formation. You have black type. And then for reverse cam, actually yung ano niya dito class is positive reaction niya is bow tie. Kita niyo to? This is the bow tie reaction. Okay? So para siyang bow tie class. Yan. So this is your Staphylococcus aureus. Okay? Streptococcus agalactiae, then the organism is Clostridium perfringens. Yan. Naintindihan? Yan. At kasi madami, tatlong ano na class, tatlong camp na yung na-discuss natin. Okay? So you have your, ayun, yung lumabas class. Wait lang. Wala. Okay. Yan. So, tatlong camp na yung nakita natin, class. Okay? So, again, para hindi makalimutan, balik na naman natin, class. You have your camp here. Kasi very important to class. Our camp. Then you have your, this one is S, Aga, Listeria mono, and Clostridium perfringens. Then this one is your Sao. This one is your Rhodococcus equi. This one is S. Aga. Okay. So this one is your organism S. Aga. This one is Listeria monocytogenes. This one is Clostridium perfringens. For the positive result naman, class, you have your arrowhead formation. Okay, you have black type. And then you have your bow tie. Okay, so yun yung difference ng cam-cam at saka r-camp natin. Okay? So, I hope na hindi natin ito makalimutan, class. Okay? So, with the reverse camp naman, class, okay, the positive result is bow tie zone of enhanced hemolysis and only your Clostridium perfringens is positive. The rest of your Clostridium is negative already. Next, biochemical tool that we can use for the identification of your Clostridium perfringens is with your litmus milk. For your litmus milk naman, class, this will determine an organism ability, of course, to metabolize your litmus milk. Okay, so meron tayong apat na possible na result. First is with the white production. If you can observe here, meron siyang white class here. Meron siyang white, ibig sabihin the litmus was reduced. Second is with the pink, then the lactose was fermented. Okay? Then bluish naman siya class, although wala siya here, the pH is still basic and lactose was not fermented. Then brown ring, kasi yung protein in the milk was utilized. Kaso wala din tayong ring here. Tapos your Clostridium perfringens can actually produce this type of production class. Acid with stormy clot formation. Okay, tadan, that one is your stormy clot formation class for your Clostridium perfringens. Red alert tayo class 2019 board exam. Okay, yung collection daw na kailangan natin for your blood is 3. Okay, 3 blood collection yung kailangan natin for Clostridium class. 3 blood class. Collection of 3 blood cultures from 3 different venipuncture site. Tapos pag stool yung sample mo, dapat 25 grams at saka 25 ml. Ganito class, every time na kailangan ng sample ng stool, tapos Clostridium or anaerobic, 25 talaga yan siya. Even with the food, 25 grams or 25 ml. Then specimen should be gram stained, cultured and isolated. Then the isolate should be serologically typed. Yun yung mga sample consideration natin with your Clostridium perfringens. For the virulence factor, you have your alpha toxin, enterotoxin, at saka beta toxin. Sorry class, that's beta toxin. For the alpha toxin naman class, mediates the disruption of your host cell membrane, causing a necrosis. You have your enterotoxin, of course, this will disrupt your mucosal cell. And then your beta toxin naman class is cytotoxic. The spectrum of disease is you have your gas gangrene. Take note with this one class because your gas gangrene is also known as your ano, ito ba yung hindi pala ito class. You have your gas gangrene or myonecrosis or necrotizing fasciitis naman sa class. Okay? And since they have a virulence factor of enterotoxin class, syempre, they will produce a food poisoning. Okay? Caused by release of your toxin after ingestion. Ito pala class, enteratitis necroticans or necrotizing enteratitis. Okay? Caused by ischemic necrosis of the jejunum. So with this one, pag may sample tayo class here, what you can do is with the wound exudate naman siya. Pwede din tayong gumamit ng capillary tube class. Capillary tube, tapos itutusok natin yan, tapos yung tissue papasok here, then we put that one on that tube and then process anaerobically. Next is you have your Clostridium botulinum class. For the culture characteristic of your botulinum, tetany, and difficile, they have a negligible characteristic on your 5% blood agar. So, ibig sabihin class, very less likely na gagamitin natin yung blood agar plate for the differentiation of the three organisms. Pero, pwede natin gamitin yung hemolytic pattern class. Kasi yung hemolytic pattern ni Clostridium botulinum in your blood agar class is beta hemolysis. Although your Clostridium tetani also produce a beta hemolysis pero narrow zone siya class. Okay? Narrow zone siya. Okay? With your Clostridium difficile naman class, yung isa sa mga pinakasikat na culture media na you have your CCFA or cycloserine, cefoxitin, fructose agar. And then, take note with this one class, ito yung CCFA. If you can observe on the picture, they will produce a yellow ground glass colonies. Yan. Red alert naman tayo dito class that the other production of your Clostridium difficile in your CCFA will produce a horse or elephant manure odor. So, para daw siyang tae ng kabayo class in or elephant. Okay? For the hemolytic pattern naman class, compared with the Clostridium tetani and botulinum, they will produce a gamma hemolysis. For the staining characteristic naman class, ito yung mga tatandaan natin with the class? Orientation of the spore. For Clostridium botulinum class, subterminal sila here, subterminal. Kung subterminal sila class, they will produce a tennis racket. Okay? Tennis racket naman siya. For your Clostridium tetani, they are oval or terminal. Kaya nagkakaroon tayo ng drumstick. Okay? Tapos, you have your Clostridium difficile, oval at saka terminal din. Okay? So if you can observe here, pag subterminal, para siyang tennis racket sa class. Kasi, pag ganito yung itsura niya, pag subterminal. Diba? So you have your spore here, subterminal. Yan. Tapos for ito sub. Pag terminal naman class, is nasa end talaga siya. Yan. Yung itsura niya. Okay? Yung neck kasi na part, pag subterminal, is pa-side, pag ganun. Pag terminal talaga is on the, pabilog talaga siya at the end. Kaya nagkakaroon tayo ng drumstick appearance. Okay? So, red alert tayo, class, board exam question. Specialized media to induce your sporulation in your Clostridia. Meron tayong tatlong culture media. So, you have your elders, medium of Duncans, and strong, at saka medium of Phillips. Kaya take note with the 3 culture media, kung lalabas sa board exam, at least may alam kang tatlo. Okay? Tapos board exam question 2019 naman class, kaya tinanong sa akin to ng estudyante ko, kay sir, para saan daw yung ethanolic shock method? So for your ethanolic shock method class, this is used for the germination of your Clostridium difficile spores. So please take note with this one, baka kasi ibalik yung tanong, okay, you have your ethanolic shock method for your Clostridium difficile spores. Additional parameter to identify your Clostridium botulinum class, you have your lipase test. Lipase test is, same with your egg yolk agar. And then what you're gonna do, class, is we allow the organism to produce the lipase that will hydrolyze your fat. And then, pag na-release, meron tayong dalawang form. You have your glycerol at saka you have your, class. You have your glycerol at saka you have your fatty acid. So the production of, kasi di ba may fatty acids na siya class. Kung may production of your fatty acid class, nagkakaroon tayo ng oil on water na characteristic. So ganito na yung itsura ni class. Okay, so we allow again the bacteria to produce the enzyme lipase that will degrade your fat or free fats. Okay? And then the end product is glycerol at saka fatty acid. For your Clostridium botulinum naman class, take note of this one. This is listed by the CDC as a potential agent of bioterrorism. And that includes your correct class. You have your Bacillus anthracis. For the virulence factor, of course, bakit siya naging virulence class? Because your Clostridium botulinum will produce your neurotoxin or BONT. It prevents the release of your neurotransmitter and then have a flaccid paralysis. Ito naman yung pinaka-common class. Flaccid paralysis, you have your Clostridium botulinum class. Okay, your Clostridium, but take note with this one. Bakit? Kasi your neurotoxin class, your botulinum neurotoxin is not only solely produced by your Clostridium difficile. Sino-sino yung mga nagpo-produce pa? You have your Clostridium botulicum, you have Clostridium barati at sa Clostridium argentinense. Take note with this one kasi pagdating sa pre-board exam, like for example, which of the following will not produce your neurotoxin? Okay, dapat alam natin yung apat. Clostridium botulinum, butyricum, you have your, class. Barati at sa Clostridium argentinense. Then for the spectrum of disease and infection class, take note with this one, food-borne botulism, infant botulism, at saka wound botulism. Okay, so take note with the food botulism class kasi yung pinaka-associated natin here, wait lang class, is class? Honey. Sulat ko class ha. So with your food botulism class, this is associated with your honey naman class. Yan. And then infant botulism, and then wound botulism naman class, at saka intestinal botulism. So lastly, we have your Clostridium tetani and Clostridium difficile class. Okay, for the two, let's start with your tetani. For your Clostridium tetani naman class, kung merong neurotoxin si botulinum, meron ding tetanoplasmin, spasmin si Clostridium tetani, a neurotoxic exotoxin that disrupt the nerve impulse to your muscle. Kaya kung meron tayong flaccid class, kanina 'di ba flaccid yung paralysis natin. Ito naman class rigid. Okay? Kung kanina 'di ba flaccid, ngayon rigid. Okay, rigid paralysis. Like for example, lockjaw, ayan, can organism establish a wound infection and then elaborates your tetanus plasmin. Tapos, binablock niya yung class? neuron receptors. Kaya nagkakaroon tayo ng lockjaw. And if you can observe also in the picture, ayan, rigidity of the body is referred to as, sorry class, yung rigidity ng body natin is referred to as your opestonus at saka rhizus sardonicus. Lastly, with your Clostridium difficile naman class, you have your toxin A at saka toxin B. Your toxin A is associated with your enterotoxin. Your toxin B is associated with your cytotoxin. Always remember class that your GTP again will, if your GTP will increase, the toxin glycosylated guanine triphosphate GTP signals the protein leading to the cell death. So if there will be accumulation of your GTP, then it will affect the functioning of your cell cake causing death. And tadan, you have your pseudomembranous colitis. Kasi di ba kanina, you have your pseudomembranous pharyngitis, di ba? Eto, sino nga yung causative agent dito, class? Pseudomembranous pharyngitis. You have your tama di ba? You have your, sorry class, you have your Corynebacterium diphtheriae. Diba? Ito yung causative agent niya. Pag naman class, pseudomembranous naman, pseudomembranous colitis. Ang naman natin yan, ang causative agent naman natin yan class is your Clostridium difficile. Bukas, meron pang isa class. Meron tayong antibiotic-associated pseudomembranous colitis. Okay, yung pangatlong merong pseudomembranous. Okay, so at least dalawa na yung na-cover natin ngayon. Okay? And we are done with your topic 1 and your topic 2 class. I know na masyadong heavy yung topic natin, specifically with your micro. Siyempre, di ba, imagine yung 1 sem natin na micro, ginawa lang nating 1 day. Di ba? 2 days. Kasi, parasitology tayo more on the 3rd day at saka mycology at saka virology. So, I know that you are tired, class, and then, just a point of motivation, kasi lahat naman tayo napapagod. It's a good sign. Pag pagod ka, class, it means that you're giving tawag nito, effort. Pag hindi ka papagod, class, ibig sabihin, hindi mo pa binibigay yung 100%. Okay? So, always be blessful na pagod ka. Ibig sabihin, you are trying your best. Okay? And then, trying your best will always have a fruitful result, class. And always take your seniors as a, tawag nito, as a motivation. I was also able to impart my expertise with them. Then they're very, class, tawag nito, very brilliant student. And I believe that you also can do what they did in the board exam. So I hope and I pray that, class, tawag nito, you somewhat gain something today with topic 1 and topic 2 lecture. Thank you. Ayun. Any questions, clarifications? Okay lang ba yung review? Feeling ko hindi. Okay lang ba?
Yes po, sir.
Yes po, sir.
Yes
Sure.